Hello everyone, I'm Dr. Arpita De. Today we are going to discuss about polymerase chain reaction. Polymerase chain reaction is abbreviated as PCR. It was invented by Carey Mullis and his colleagues in the year 1983, and for this great milestone invention he was awarded with the Nobel Prize. PCR technique is an in vitro technique that allows amplification of specific DNA sequences or target sequences selectively millionfold in just a few hours. That is, the target sequence, no matter how less it is, that paltry or that meager amount of DNA is amplified in a very short span of time. This brief flow chart summarizes the entire process of PCR. Genomic DNA is extracted. Usually small sample of genomic DNA is used, and then one section, that is the preferred or the target sequence, is replicated many times. That gives millions of copies of the DNA of interest. Now coming to the principle of PCR. The target sequence of DNA is denatured and amplified selectively with the help of specific primers and enzyme to get multiple copies of the sequence of interest. Now to run the PCR, we need certain specific components. Fine. First of all, we need DNA sample. Then the primers. These primers include both the reverse primer as well as the forward primers. Then the nucleotides or DNTPs, Taq polymerase, obtained from Thermus aquaticus. Since it can withstand a very high temperature, this DNA polymerase is preferentially used for PCR. Then we have buffer, magnesium chloride. All these are chief components of PCR. Now coming to the PCR master mix. It is a pre-mixed solution containing Taq polymerase, DNTPs, magnesium chloride, reverse and forward primer, template DNA, nuclease-free water and buffer. Conventional PCR involves 25 to 50 repetitive cycles, with each cycle comprising of 3 sequential reactions. First is extraction and denaturation of DNA. Second, primer annealing to single-stranded DNA. And third, extension of primer target duplex. Coming to the first step, that is denaturation. The reaction mix is heated to 95 degrees Celsius for 15 to 30 seconds to denature the target DNA into single-stranded DNA that serves as template for DNA synthesis. If you carefully look into this graph, you can see this A marks the denaturation condition, which is done at 95 degrees Celsius, followed by cooling, that is the temperature for primer annealing, which is usually maintained at 55 degrees Celsius. Then again the mixture is heated, that is the temperature is increased to about 72 degrees Celsius, where extension occurs. Step number 2 is primer annealing. The mixture is rapidly cooled to allow the primers to bind to the sequences on each of the 2 strands flanking the target DNA. The primer should only bind to the desired DNA sequence. This should be taken care of. Only one primer binds to each strand. Primer annealing is followed by DNA synthesis that occurs at the 3' OH end of each primer. Primers are extended by joining the bases complementary to DNA strands, and remember, primer annealing temperature is 55 degree. This temperature has to be maintained. That denaturation occurred at a very high temperature, so this rapid cooling has to be achieved to allow primer annealing, otherwise it won't happen. Now the last step, that is extension. The temperature of the mixture is raised to 72 to 75 degrees Celsius to allow the DNA polymerase to elongate each primer by copying the single-stranded templates with the help of Taq polymerase. Right? The enzyme is capable of withstanding the denaturing temperature of 94 to 95 degrees Celsius. If you refer to the figure, you can see that. So the above three steps are repeated in PCR cycle. Fine. Now after the amplification of DNA is over, the amplified DNA is analyzed by agarose gel electrophoresis followed by UV visualization. These are some of the very important applications of PCR. PCR is widely used in the field of medical research, forensic science. So please go through these applications. PCR is such a word, it has become very famous during the COVID. Thank you so much.